BTEC Applied Science, Unit 1 Biology, synapses. Very important. We need to know quite a bit about synapses. So, what is a synapse? A synapse is the connection between two neurons. Do you remember you have your axon terminal at one end and then you've got your dendrites at the other end, and the gap between them is the synapse. So it's where a signal is passed from one neuron to another neuron. Your brain is full of neurons and they're all kind of sending lots and lots of signals to, not to each other, not to every other neuron, but there's lots of synapses in your brain and in other parts of your body too. Okay, so there's a synapse. There's electrical synapses and chemical ones. We are only interested in chemical synapses where the signal is passed from one neuron to another using a chemical called a neurotransmitter. And we'll talk about neurotransmitters quite a lot in the next video. Here's a diagram of a synapse, and we're going to be able to label this very shortly. You see the action potential comes down. Remember from a previous video what an action potential is. This voltage signal comes down and that triggers several things are going to happen. Chemicals are going to go through the gap through the synaptic cleft, the gap between the two neurons. So you've got your pre-synaptic neuron and your post-synaptic neuron, before and after. Okay? You've got inside the presynaptic neuron, there are vesicles. Remember that vesicles are like little taxis and they carry the neurotransmitter. So there's the chemical called the neurotransmitter inside the vesicles. Now, what happens is this. The action potential comes down and it opens up voltage-gated calcium channels. So these calcium channels open up and calcium ions come into the presynaptic neuron. Calcium ions come in, and what happens is that enables the vesicles to attach themselves to the presynaptic cleft. Okay? The action potential comes down, gates open, allowing calcium ions to enter. So calcium ions go in. This activates docking proteins on the vesicles. So now the vesicles can attach themselves to the presynaptic cleft. And then when that happens, the neurotransmitter can come out of the vesicle, travel across the gap. The vesicles release their neurotransmitters into the cleft. For example, acetylcholine is an example of a neurotransmitter, acetylcholine. What happens next is that the neurotransmitters dock with receptors attached to the gates on the other side. The receptors open. They only stay attached for a few milliseconds. After this, they pop off. But what happens while they are open is that sodium ions can rush in, and that creates the action potential, which then carries down through the second neuron. When these chemically gated channels open, this allows the flow of sodium ions, which causes an action potential in the postsynaptic neuron. So learn that sequence of events. Go through the video several times, make some notes, learn that sequence of events. Memory is a set of neural pathways. Now, what does that mean? That basically means if a synapse is fired again and again and again, look at what difference it makes. Can you see what difference it makes on the diagram? Basically, there are more receptors. Okay, so the link between them is more established. The synapse becomes more established. And that basically means, well, that's what memory is. Okay, that sticks, that link between the synapses. What's the capital of Japan? Tokyo. There you go. It's more established. Here are the key words that you should know the meaning of. The synapse is the link between the two neurons. A neuron is just a nerve cell. Chemical synapses, that's what we're interested in, not electrical ones. You've got your presynaptic neuron and your postsynaptic neuron, kind of before and after. The voltage-gated calcium channels, which open up, the action potential causes them to open up. The synaptic cleft is the gap that we're talking about between the two neurons. The presynaptic vesicles. So vesicles are like little taxis and they carry the neurotransmitter inside the presynapse. Acetylcholine is an important neurotransmitter. Docking proteins are on the presynaptic cleft. Neurotransmitter is the chemical that crosses the gap. Receptors on the postsynaptic cleft, and they open up, and then sodium ions can rush in and create the action potential, which carries the signal down the second neuron. Here are some interesting facts. Your brain has about 100 billion neurons. And each neuron has about 1,000 to 10,000 synapses. There's an awful lot of connections in your brain. Synapses can strengthen or weaken over time depending on how much they are used. Remember that when you're revising for your Unit 1 exam. The synaptic cleft is less than 5 millionths of a centimetre wide. And many drugs, we'll talk a lot about this in the next video, many drugs work by interfering with the function of neurotransmitters. And several diseases are caused when neurotransmitters aren't doing their job properly, as we shall see. There's a little bit of information about acetylcholine for you. Read it. Here are some questions which you should definitely be able to do. Describe in detail how an action potential is passed across a synapse. Include a label diagram in your answer. 6 marks, lots of information. What happens to the neurotransmitters in the synaptic cleft after they have done their job? And if you want to do well in the exam, you should revise. Can you give a biological reason why you should revise?